We know that immune therapies have just been transformative for many types of cancer, but specifically for kidney cancer, they sort of reawaken the immune system. But unfortunately, there's this tremendous unmet clinical need from patients who receive an immune therapy, but it's not effective, and they receive another standard care and it's not effective. And so developing new immune therapies, hitting new targets is incredibly important and has not been something that's been particularly successful thus far in kidney cancer. And so, with that background, there's an interesting target that comes out of preclinical studies called HPK1. It's essentially a brake on T cells, but located within the cell itself. It's a molecule that basically stops T cells from becoming activated once it sees its target. So, the hypothesis going into this study was we know from preclinical work that blocking HPK1 really improves anti-tumor immunity, can help to clear tumors. In a first-in-human study, if we can pharmacologically inhibit HPK1, would that lead to anti-tumor responses? So this was a phase one first-in-human study of this molecule NDI-101150, a very selective HPK1 inhibitor. It started with a dose escalation, classic 3+3 design to find the maximum tolerated dose. And then there was a dose expansion in a few cancer types, including kidney cancer, gastric and gastroesophageal tumors, and non-small cell lung cancer. What we saw was that this was safe, that the toxicity profile was relatively minimal, not high grade autoimmune effects, manageable GI toxicities. And I think most notably, it really did have single agent activity, clinical activity within kidney cancer. So the goal is really to see how this might translate into new therapies for patients. And so we know from the responses within kidney cancer, where in a very treatment refractory setting, after anti-PD-1 therapy, after anti-angiogenic therapy, we saw a patient who had a complete response to this therapy, resolution of their tumor, two patients with partial responses, and three patients with stable disease that lasted over 6 months. In one case, a stable disease that lasted for over 2 years. And so I think the next step is to really examine this as a much larger group of patients. We now have the proof of concept. We know that this has a biological plausibility from our preclinical studies and some of our translational work. We know that the signals of clinical activity and the signs of safety. Now we need to study this in a much larger group of patients with clear cell kidney cancer and really determine what are the efficacy parameters.